All cells are composed of organic molecules. Here are a few examples of how organic molecules interact and react to form new molecules. The hydrolysis of starch into glucose. Here, starch hydrolyzes or gains a water molecule, splitting it apart into two glucose molecules. The hydrogen from the water attaches to one monomer, while the hydroxyl group attaches to the adjacent monomer. Here, the substrate is starch. The active site is the bond between the two starch monomers, and the enzyme is amylase, which splits the molecule apart. Dehydration of glucose into glycogen. Here, two glucose molecules go through a dehydration reaction where they lose a water molecule to form glycogen. One molecule provides a hydrogen, and the other provides a hydroxyl group to form water molecule. Here, the substrates are the glucose molecules. The active site is where water is formed and lost, and the enzyme is glycogen synthase, which turns glucose to glycogen. Now we move on to the cells. There are two types of cells we'll be talking about, plant and animal cells. These cells are composed of various organelles. On the left, we have a plant cell. It has a cell wall which protects and supports the cell, a plasma membrane which is composed of the phospholipid bilayer and the proteins that regulate what moves in and out of the cell, a central vacuole that supports the cell and stores materials, a smooth and rough endoplasmic reticulum which transports and manufactures proteins, a Golgi apparatus which transports materials throughout the cell, a chloroplast which goes through photosynthesis and produces food and energy for the cell, a peroxisome which converts fatty acids to sugar and assists with photorespiration, a mitochondria which goes through cellular respiration to produce energy for the cell by breaking down glucose and oxygen, a nucleus which contains DNA and directs and manages all cell activities, a ribosome which manufactures proteins. On the right, we have an animal cell. It has a plasma membrane, vacuoles, a smooth and rough endoplasmic reticulum, a Golgi apparatus, a peroxisome, a mitochondria, ribosomes, nucleus, a centriole, and a lysosome. The centriole forms spindle fibers to aid in cell division, and a lysosome breaks down cell waste and exports it out of the cell. DNA replication and chromosomal duplication. Organisms are formed by single cells reproducing. For this to happen, a cell's DNA has to replicate. DNA replicates in a way that we call semi-conservative replication, in which there are two copies produced that each contain one of the original strands and one new strand. Guanine is the complementary base of cytosine, and adenine is the complementary base of thymine in DNA and of uracil in RNA. Gene duplication, or chromosomal duplication or gene amplification, is a major mechanism through which new genetic material is generated during the molecular evolution. In mammals, meiosis produces the cells that combine and form a zygote, which goes through mitosis to grow and develop. Meiosis is the root of all genetic diversity, and here's how. Crossing over is the process where homologous chromosomes pair up with each other and exchange different segments of their genetic material to form recombinant chromosomes. Non-disjunction is the failure of homologous chromosomes or sister chromatids to separate properly during cell division. Independent assortment is something that Mendel came up with. He came up with the law of independent assortment, which states that alleles of different genes assort independently from one another during gamete formation. Segregation states that during the production of gametes, the two copies of each hereditary factor segregate so that offspring acquire one factor from each parent. Random fertilization. This means that the collection of genes within one gamete, such as a sperm cell, do not give that gamete a lesser or better chance than any of the other sperm cells of fusing with an egg cell to produce a zygote. On to mitosis. G1 is the first growth period of the cell cycle, during the interphase in which the cell grows and cytoplasmic organelles are replicated. S. During this phase, DNA is replicated. G2. At the G2 checkpoint, the cell checks two things. First, it checks DNA for any damage that might have occurred during replication. It also ensures that all the proteins needed for cell division are present. There is an acronym, PMAT, prophase, metaphase, anaphase, telophase. Prophase is when chromosomes become visible as paired chromatids, the nuclear envelope disappears. Metaphase is when the chromosomes become attached to the spindle fibers. Anaphase is when the chromosomes move away from one another to the opposite poles of the spindle. Telophase is when the chromatids or chromosomes move to the opposite ends of the cell and two nuclei are formed. Cytokinesis. A cell requires proteins to complete replication and division as well as every other activity, but how does it make them? Protein synthesis happens in two major steps, transcription and translation. Here, an insulin molecule is being transcripted. That means that its DNA is being turned into mRNA or messenger RNA. The main enzyme that accomplishes this is RNA polymerase. mRNA, before it goes through the spliceosome, is made up of a G cap, introns, exons, and a poly A tail. The spliceosome cuts out the introns and splices the exons together. This leaves the G cap and the poly A tail to protect the molecule from being broken down by hydrolyzing enzymes. Next comes translation. This involves a ribosome and its E, P, and A sites, tRNA, and proteins. In translation, a tRNA molecule bonds to the mRNA codon underneath the A site. In the P site, it is another tRNA molecule with a long string of proteins attached. There is a reaction, and all of the proteins attached to the tRNA in the P site move over, and the bottom of this strand attaches to one protein on the tRNA in the A site. Both tRNA molecules move one site to the left. The tRNA without any proteins on it is now detached from the molecule. A new tRNA comes in and attaches to the mRNA at the A site, and the process starts over again. This long chain of peptides goes through a series of complex reactions and folds into the shape of an insulin protein. This protein travels back to the rough ER, then through the Golgi apparatus, and is placed inside a vacuole. This vacuole binds to the plasma membrane and expels the protein outside of the cell. The plasma membrane controls everything that moves in and out of the cell, but how? What's it made up of, and how does it let us do that? Here we have a phospholipid. These make up the majority of the plasma membrane and are comprised of a hydrophilic polar head and hydrophobic nonpolar fatty acid tails. In animal cells, the plasma membrane is composed of the phospholipid bilayer, cholesterol molecules, and the channel transport proteins that transport or carry out materials in and out of the cell. Here we have water on the right entering the cell through its specialized channel protein called an aquaporin. This is called osmosis, which is a type of passive diffusion. Here we also have oxygen and carbon dioxide diffusing into the cell. They are both small nonpolar molecules, so they are able to diffuse simply across the cell membrane without channel or carrier proteins. Next we have the sodium-potassium pump. Here's how it works. Sodium and potassium plus ATPase holds 3 spots for sodium ions. They bond to each of these sites. ATP is converted to ADP, releasing energy that powers the enzyme to shift position. The sodium detaches from sodium-potassium ATPase and it is released from the cell. The 2 potassium receptors are now exposed. 2 potassium ions attach to these 2 sites. ADP is converted to AMP and the enzyme shifts again. The potassium ions detach from the enzyme and are released into the cell. To correctly grow and develop, the cells of a multicellular organism have to communicate effectively and efficiently. Here's how they do it. Here, a ligand is attaching to a receptor protein on the membrane of a muscle cell. The 3 steps of a cell communication pathway are reception, transduction, and response. In reception, a target cell detects a signaling molecule. During transduction, a bound signaling molecule changes the receptor protein, and the signal is converted to a cellular response. During response, the cellular response happens. All of this communication, regulation, division requires energy. Plant cells make their own food and then break it down into energy through photosynthesis. The formula for photosynthesis is 6CO2 plus 6H2O equals C6H12O6 plus 6O2. Each stoma is surrounded by 2 guard cells which can open and close depending on environmental conditions. When moisture is plentiful, the guard cells swell with water, forcing the opening of the stoma open and allowing gas exchange to occur. The xylem takes water from the roots and moves it upward. The phloem transports food from the leaves to the rest of the plant. The chloroplast has stacks of thylakoid known as granum that are situated in the stroma. The light-dependent reactions, water to oxygen gas, happen on the thylakoids. The Calvin cycle, CO2 to water, happens in the stroma. Animal cells lack the capacities to perform photosynthesis, so they undergo cellular respiration to produce energy. The formula for cellular respiration is C6H12O6 plus 6O2 equals 6H2O plus 6CO2 plus ATP. This is the opposite of photosynthesis. The mitochondria contains the cristae and matrix covered by the inner and outer membrane. Glycolysis turns glucose into pyruvate, and this takes place in the cytoplasm. The Krebs cycle occurs in the mitochondrial matrix and generates a pool of chemical energy. from the oxidation of pyruvate, the end product of glycolysis. Oxidative phosphorylation takes place in the inner mitochondrial membrane in contrast with most of the reactions of the citric acid cycle and fatty acid oxidation, which take place in the matrix. Lactic acid fermentation is the process by which our muscle cells deal with pyruvate during anaerobic respiration.